Topic 1: Biological molecules. Explain role of inorganic ions in plants. Inorganic ions can be used to make DNA and amino acids, to strengthen the middle lamella, to produce chlorophyll, and to make ADP and ATP. Making DNA means that you can control the cell activities. Making amino acids means that proteins can be synthesized. Strengthening the middle lamella holds the cell walls together. Producing chlorophyll means that light energy can be absorbed for photosynthesis, and ATP can be used as a readily available source of energy. To make DNA and amino acids requires nitrate ions, and that helps to control the cell activities and synthesize proteins. Calcium ions are used to form calcium pactate, and this strengthens the middle lamella to hold the cell walls together. Magnesium ions are used to produce chlorophyll so that light energy can be absorbed for photosynthesis. And phosphate ions are used to make ADP and ATP, which are the readily available sources of energy. Explain properties of water. Water contains one oxygen atom and two hydrogen atoms. The hydrogen atoms are slightly positive, whereas the oxygen atoms is slightly negative. This means that one water molecule is polar or dipolar because of the fact that the side that contains the oxygen is slightly negative and the other side of the water molecule that contains hydrogen atoms is slightly positive due to the uneven sharing of electrons during the covalent bonding. Therefore, hydrogen bonds can form between the hydrogen atom of one water molecule and the oxygen atom of an adjacent water molecule, causing an attraction between water molecules. This has a significance to organisms. An example of this is in the transpiration stream in xylem, where the hydrogen bonds form between adjacent water molecules, causing them to stick together. And this is known as cohesion, where the water molecules are cohesive by sticking together due to the hydrogen bonds that form. Another example of this is the hydrogen bonds that form between water and other different charged molecules, which could be either polar or ionic, such as those in the xylem wards, and this property is known as adhesion. So water molecules are adhesive when they can form hydrogen bonds to other different charged molecules that are polar or ionic that are not water. Other significance to organisms due to the polar properties of water is the fact that water has a very high specific heat capacity. This means that you need to give it huge amounts of energy to change its temperature. So by having a high specific heat capacity, that means the temperature of water remains constant or is stable for aquatic organisms to live in. Water is also a very good polar solvent because it dissolves charged molecules that are either ionic or polar. This is useful because those dissolved solid molecules are easier to react with each other in metabolic reactions within the cytoplasm when they are dissolved. Dissolved molecules can also be transported easier because water is used as a transport medium such as blood or in the transpiration stream of xylem. Water also has a surface tension property because of the fact that water molecules are polar due to the hydrogen bonds that they form, making them cohesive between adjacent water molecules. And this results in an inward force at air-water surface because there is more hydrogen bonding between water molecules than there is between water and the air. Another property of water is that it is incompressible, and this provides support because it can be used in the hydraulic systems of certain organisms. due to the fact that it's very hard to squash a water molecule. One other property of water is that it has a maximum density at 4 degrees Celsius. Water density decreases below 4 degrees Celsius, and that causes ice to float. So for most materials, the solid is a higher density than the liquid. However, for water, below 4 degrees Celsius, solid water, which is ice, floats because it has a lower density than liquid water, and this is useful for organisms because it insulates aquatic organisms that live below the water. Describe structure of glucose and ribose. These are both types of monosaccharides. Glucose is a hexosugar because it contains 6 carbon atoms, whereas ribose is a pentasugar because it contains 5 carbon atoms. The general formula of a monosaccharide is CH2O and then N number of carbon atoms. Therefore, there is twice as many hydrogen atoms as carbon atoms, and the number of oxygen atoms is the same as carbon. So for instance, for hexosugars, because there's 6 carbon atoms, the formula would be C6H12O6. So double the number of hydrogen atoms and same number of oxygen atoms as carbon. For ribose, which is a pentasugar, it contains 5 carbon atoms, so the number of hydrogen atoms should be 10, and therefore oxygen will be 5. So the formula for ribose is C5H10O5. There are 2 types of glucose, alpha glucose and beta glucose. In alpha glucose, the hydroxyl group, OH, is below the ring or below the plane of the sugar ring. Whereas in beta glucose, that hydroxyl group is above the ring or above the plane. Deoxyribose is also a pentasugar, and it is different to ribose because when you look at carbon 2, the oxygen in the hydroxyl is replaced by a hydrogen atom. So in other words, there is no oxygen atom, there's just hydrogen on its own. Here we can see an example of 2 monosaccharides that are bonding with each other. Explain how disaccharides and polysaccharides are formed and can be split. So disaccharides are made up of 2 monosaccharides that are joined together by a glycosidic bond. You can see in the example 2 alpha glucose bond together, and not only do they form a disaccharide, which is maltose, a water molecule is also formed. So that water molecule is removed from the 2 monosaccharides. So we call this a condensation reaction. The glycosidic bond that forms between the 2 alpha glucose is a 1,4 glycosidic bond because it forms between the carbon 1 of one monosaccharide, one of the alpha glucose, joined to the carbon 4 of another. There is an opposite reaction to condensation, and this is where water molecule is added to the glycosidic bond. So when you add water to a disaccharide, when you're adding it to the glycosidic bond, this is known as a hydrolysis reaction. And that's because you are splitting the 2 saccharides by breaking the glycosidic bond. So hydro means water, lysis means split. So using water or adding water to split the glycosidic bond, and therefore 2 saccharides, a disaccharide, can be split to form 2 monosaccharides. And the same can be done with polysaccharides to form individual monosaccharides or smaller saccharide molecules. Example of this are these irreversible reactions between disaccharides and 2 monosaccharides. So maltose can be hydrolyzed to form 2-alpha-glucose, or the reversible reaction where 2-alpha-glucose can be reacted together in a condensation reaction to form maltose. Lactose can be hydrolyzed to form alpha-glucose and beta-galactose, which is a different type of sugar. And again, you've got the reversible reaction where you can do a condensation reaction to join these 2 monosaccharides together to make the disaccharide lactose. The disaccharide sucrose can be hydrolyzed to form the 2 monosaccharides alpha-glucose and fructose. And again, you've got the reversible reaction where you can join these 2 monosaccharides together in a condensation reaction to form the disaccharide sucrose. And this is the example we saw earlier. We've got 2 alpha-glucose here where they are reacting with each other. The monosaccharides react with each other to form a disaccharide called maltose, and to do this, a condensation reaction occurs where you also make a water molecule, and the 2 monosaccharides form a glycosidic bond, which is known as a 1,4 glycosidic bond because it happens between carbon 1 of one of the alpha glucose molecule and carbon 4 of the other alpha glucose molecule. Explain how polysaccharide structures relate to their function. Here we've got 2 examples of polysaccharides, amylose and amylopectin. So these are polysaccharides because you've got lots of repeating units of saccharides joined together for a condensation reaction where they're bonding to each other with glycosidic bonds. And you can see that amylose and amylopectin are slightly different to each other, because amylose has just got a straight chain of saccharide with 1,4 glycosidic bonds, whereas the amylopectin has got additional branches, and these branches are joined to each other through a 1,6 glycosidic bond. Here's another example of another polysaccharide, but here the polysaccharides are forming cross links with each other through hydrogen bonds. So the different types of polysaccharides you need to know about are starch, which is a mixture of the amylose and amylopectin. Then we've also got glycogen, which is found in animals, and cellulose in plants. Now if you look at the starch, the 2 different types of polysaccharides that make the starch, the amylose in the starch is unbranched. It's an unbranched polymer containing alpha-glucose molecules joined together by 1,4 glycosidic bonds. And this polymer or chain forms a spiral shape. That helical spiral shape is due to hydrogen bonds with each other, and that increases the compact storage of energy. So in other words, you could fit more glucose molecule by spiraling through the formation of hydrogen bonds. Starch also contains amylopectin, and these amylopectin has branch polymer of alpha-glucose molecules joined by both 1,4 and 1,6 glycosidic bonds. And those 1,6 glycosidic bonds or multi-branch polymers provide rapid hydrolysis. So in other words, you can break those 1,6 glycosidic bonds by hydrolyzing them, and that releases glucose molecules that could then be used to do more respiration so that more energy can be released. Glycogen is a polysaccharide found in animals, and it has the same structure as amylopectin in the fact that it is a branch polymer of alpha-glucose joined by 1,4 and 1,6 glycosidic bonds. It has more branches than amylopectin, therefore you could do more rapid hydrolysis of these multi-branch polymers to release more energy because more glucose is available, and that is because you could do more respiration. Cellulose is different because just like amylose is an unbranched polymer, but however it is made of beta-glucose molecules that are joined together by 1,4 glycosidic bonds. And because of the fact that it's made up of beta glucose, therefore it could form cross links between the long straight chains by hydrogen bonds, and this provides additional strength in the cell wall. Both starch and glycogen are very compact energy storage molecules, and that is their main function. All three of these polymers, starch, glycogen, and cellulose, are very large insoluble molecules. So insoluble means that they don't dissolve in water, and because they don't dissolve in water, that means they will not leave the cells. They will stay in the cells, which makes it very easy for them to be stored or stay as a structure in cellulose case. And this means that they have no impact on osmosis or solute or water potential because of the fact that they do not dissolve in water. Explain how triglyceride, which is a type of lipid, is synthesized. So here you can see an example that lipids, which are fats and oils, are made up of glycerol and fatty acids. The glycerol combines with one fatty acid to form a monoglyceride, or it can combine and bond with two fatty acids to produce a diglyceride, or three fatty acids to form a triglyceride. The reaction happens between the hydroxyl group of the glycerol and the carboxyl group of the fatty acid, and this forms a water molecule as well, which is removed, which means that it is a condensation reaction. An ester bond is formed in that condensation reaction, and that forms between the oxygen and the carbon of the fatty acid. So in this case, the bond between the glycerol and the fatty acids is known as an ester bond. The fatty acids that are bonded to the glycerol can be different to each other because of the hydrocarbon chain that they are made of. So here's an example of a triglyceride being made. You can see it's made of one glycerol and three fatty acids, and it happens between the hydroxyl group of the glycerol and the carboxyl group of the fatty acid to form an ester bond, and because there are three fatty acids, there are three ester bonds formed here. Three water molecules are also made because this is a condensation reaction. Describe differences between saturated and unsaturated lipids. So here we can see a triglyceride where we've got a glycerol and three fatty acids bonded to each other. And lipids can be different to each other due to their hydrocarbon chain. So here a saturated fatty acid contains only a single covalent bond between the carbon atoms in the hydrocarbon chain. And that hydrocarbon chain is all saturated with hydrogen atoms. In other words, they are bonded with hydrogen atoms. You cannot fit any more atoms into that hydrocarbon chain. So every single carbon atom has got a single covalent bond with each other. In the middle example, you got fatty acids that contain a double or one double covalent bond within the hydrocarbon chain. So this is a monounsaturated fatty acid because of the fact that it's mono because it contains one double covalent bond, and it's unsaturated because there is a double covalent bond. In other words, there are not 100% single covalent bonds. There is one double covalent bond. So because it's double covalent, it's unsaturated, and because there's only one of them, it's monounsaturated. In a polyunsaturated fatty acid, there are more than one double covalent bond within the hydrocarbon chain. And these are the different types of fatty acids you could have that could bond to glycerol, so that you can make lots of different types of triglycerides or diglyceride or even monoglycerides based on the different types of fatty acids due to the fact that they could be saturated or unsaturated. Explain how structure and properties of. So here we can see we've got phospholipids, and how do these phospholipids relate to their function in cell membranes? So here are phospholipid, and they are part of the phospholipid bilayer. And we want to know what's a component of a phospholipid. What does a phospholipid contain? It's called phospholipid, so the phosphor means that it contains a phosphate, and the lipids, as we said earlier, contains the glycerol, and in this case there's two fatty acids because of the fact that a phosphate is bonded to one of the hydroxyl group that has been removed in the glycerol. When you look at the phospholipid bilayer, you can see that the phosphate group heads are polar, and that's because they are hydrophilic. They can interact with an aqueous environment. They are attracted to water as they form hydrogen bonds. So these polar phosphate group heads are on the outside of the phospholipid bilayer. On the inside of phospholipid bilayer, you've got the non-polar fatty acid tails. They are oriented inwards, and the reason why that is because these fatty acid tails are hydrophobic, so they repel water. Because they're non-polar, they can't form hydrogen bonds with water, so they prefer to be inwards, away from the aqueous water environment. How does the structure and properties of lipids relate to their role in nature? Well, lipids have a greater energy storage per gram than carbohydrates and proteins, and this is because there's a high percentage of carbon-hydrogen bonds, which means that when you break these bonds, you can release more energy in respiration. So therefore, lipids are a good long-term energy storage molecules. They are also waterproof properties because of the fact that the fatty acids of lipids are hydrophobic or non-polar. Therefore, they repel or can't bond to water, making the material insoluble. This helps to make organisms waterproof. So example of this is fur in mammals, or feathers in birds, or the outer surface wax in insects and plants. All of those have got lipid structures inside them that makes the organism waterproof. Lipids are also good thermal and electrical insulators. That's because water-soluble substances like charged ions can't get through the polar lipid layer. And this helps to insulate against heat loss in animals. So an example of this is thick blubber layer in whales, where you're retaining the heat inside. Also, fatty myelin sheet insulates the nerves, so electrical impulses travel faster through the neuron, and that reduces the loss of electrical energy out of the neuron. Here are examples of amino acid, and these amino acids are different to each other because they could either be non-polar, polar, or have ionic charges, which is positive or negative. Why are these amino acids different to each other? Is due to their R group. Outline structure of amino acids. All amino acids have an amino group made of NH2, a carboxyl group made of COOH, a hydrogen atom, and then opposite that, an R group, which is different. Amino acids can be joined together because they are a monomer, and they join together or are linked together when they react with each other to form a peptide bond, and this forms a polymer called polypeptide. Explain formation of polypeptides and proteins. Here's an example of two amino acids which are reacted to each other. You could see that the reaction occurs between the carboxyl group and the amino group of different amino acids to form the peptide bond. And this also results in the formation of a water molecule, which means that this is a condensation reaction. The reverse reaction could also occur where you add water to a dipeptide molecule or a polypeptide molecule, and the water molecule is then used to lyse or break the peptide bond to form the amino acid structure, and this occurs through a hydrolysis reaction. Now when we look at these amino acids, we can see that the R groups can be hydrophobic or hydrophilic. Therefore, the structure of the R group affects how the amino acid bonds with other amino acids in a protein or polypeptide, depending on whether the R group is polar or non-polar. So if it's polar, clearly it can form hydrogen bonds with each other, whereas if it's non-polar, it can't form hydrogen bonds with each other. And here's the example we saw earlier, where we see a condensation reaction happening between two amino acids, between the carboxyl group of one of the amino acid and the amino group of another amino acid to form a peptide bond with the release or the removal of one water molecule in the formation of a dipeptide. And as we said earlier, all of these R groups in the 20 different types of amino acids can be different to each other, therefore making them polar, non-polar, or ionic. Now, when making a protein, there are different protein structures. You could have primary, secondary, tertiary, or quaternary structures. And the primary structure we can see here's the polypeptide as a straight chain. In the secondary, we can see that it can form shapes. In tertiary, we see additional folding. And in quaternary, we can see that there's more than one polypeptide. Describe role of bonding in protein structures. So here's the example we saw earlier with amino acids bonded to each other to form peptide bonds in a condensation reaction. So this is known as the primary structure, which is just a sequence of amino acids that makes up the polypeptide chain, and they're clearly held together by peptide bonds. The secondary structure is when you're folding the primary structure of the polypeptide chain into two different types of structures. They could be either an alpha helix or a beta pleated sheet, and this is because there is hydrogen bonds between the amino acids that make up the polypeptide structure. In a tertiary structure, you've got now the additional 3D shape due to the folding of the protein, and these are held together by either ionic, hydrogen, and or disulfide bonds between the R groups of the amino acids that make up the protein. And then in a quaternary structure, this is where polypeptides that are folded up into either an alpha helix or beta pleated sheet arrangement with, in this case, two or more polypeptides. And these two or more polypeptides are joined together by hydrogen bonds, disulfide bridge, and can also be ionic as well. So as we mentioned earlier, there are 4 different types of protein structures: primary, secondary, tertiary, and quaternary. We know in primary structure is just the polypeptide chain formed by amino acids bonding to each other by peptide bonds. In secondary structure, we've got additional folding due to hydrogen bonds into alpha or helix or beta pleated sheet. And then tertiary structure, we've got the 3D shape due to folding by ionic bonding, hydrogen bonding, and disulfide bridges between the R groups. And then in quaternary, there is just those same exact bonds, but this time forming between more than one polypeptide. Explain significance of protein structures in determining properties of fibrous and globular proteins. So here we could see two different types of protein structures, one of them fibrous and one of them globular. The collagen is fibrous and the hemoglobin is a globular protein. And when we look at the difference between both of them, you can see that the fibrous protein has little or no tertiary structure, whereas a globular protein does have a tertiary or 3D and can have quaternary structures that are held by hydrogen and disulfide bonds. The fibrous structure, which is the collagen, is made of long polypeptide chains which have cross linkages between them, whereas the globular protein, hemoglobin in this case, is folded into a globular shape. The fibrous protein is insoluble in water, which is because it has a structural property, so collagen can be found in examples such as the skin and hair and nails. Whereas globular protein are soluble in water because the globular proteins are folded so that the hydrophilic groups, so those R groups that have got hydrophilic properties, most likely they are polar, they are on the outside of the protein, and the hydrophobic, such as the non-polar R groups, are on the inside. So by having the R groups that are polar or ionic exposed on the outside, they can form hydrogen bonds with water because water is a polar solvent, so therefore they can form hydrogen bonds with water and become dissolved in water, making them soluble. Explain how the structure of collagen and hemoglobin are related to their function. So due to the way that these proteins are structured, they therefore have an impact on the function of the protein. So this is collagen here, and you can see it's actually made up of 3 different polypeptides. But these polypeptides are forming cross linkages with each other, which is the hydrogen bonds. And in collagen, they are mainly made of glycine or very small amino acid groups. And this enables the collagen fibers to be much closer together. This allows formation of bonds that hold the polypeptide chains together, making collagen strong. It's made of 3 polypeptide chains held together by lots and lots of hydrogen bonds in the cross linkages. It's also flexible because of kinks in the protein chain. Whereas hemoglobin is a quaternary structure made up of 4 polypeptide chains, 2 alpha and 2 beta, and each of those polypeptide chains have an iron-heme prosthetic group attached to it. Describe structure of DNA and RNA. So here's our DNA, and from GCSE we would have learnt some of these structures previously. So DNA is made up of a repeating monomer structure. The DNA nucleotide is composed of one nitrogenous base. That could be G, C, A, or T, or in RNA it could also be U. It's also made up of one pentose sugar, and it's made, which could be deoxyribose in DNA or ribose in RNA. And a DNA nucleotide is also made up of at least one phosphate group. So one nitrogenous base, one pentose sugar, and at least one phosphate group. And these nucleotide monomers are held together by a phosphodiester bond, which happens between the pentose sugar on one nucleotide and the phosphate group of the adjacent nucleotide. The nucleotide monomers are linked together by a condensation reaction, and that produces the whole large DNA polymer or RNA polymer or the polynucleotide strand or chain. In DNA, there are two sugar phosphate backbones, or DNA strands, or polynucleotide chains, that form the double helix structure. And these backbones or chains are linked to each other because there is base pair between nitrogenous bases that are complementary. So remembering GCAT, you can see that you've got guanine and cytosine that could complementary base pair with each other, and adenine and thymine can complementary base pair with each other. And these bonds between them are held together by hydrogen bonds. Now both cytosine and thymine are pyridine bases, whereas guanine and adenine are purine bases. So the way to remember that is cytosine and thymine have Y in their spelling, just like pyridine, as guanine and adenine are purine bases. Now in RNA, the thymine is replaced by uracil, so therefore uracil in RNA is also a pyridine base. Now when we look at the DNA structure, it has a 5' end, and this is the end where there's the carbon 5, and the other end of the strand is the 3' end, where you've got the carbon 3 sticking out. When we look at the other strand, or the opposite strand, the 5' end is at the opposite side. That means that the two strands are antiparallel. They run in opposite directions to each other. That's really important if you have to draw the structure of DNA. And you could notice this because you could look at the shape of the ribose sugar and you could see that the 5' carbon end on one of the strands is at the opposite to the 5' end of the other strand. So they run in antiparallel opposite directions. So those are the big biomolecule building blocks that we learnt about: nucleic acids, which are DNA and RNA, made up of nucleotides as the monomer; carbohydrates, made up of monosaccharides that can be joined together in glycosidic bonds. We've got lipids, which are made up of glycerol and either 1, 2, or 3 fatty acids that can be different due to them being saturated or mono or polyunsaturated. And then you've also got proteins, which have got amino acid monomers linked to each other by peptide bonds to form polypeptide polymers. And all these reactions, when you join the single units with each other, you're forming condensation reactions with water being released, or you could break these bonds through hydrolysis by adding water. Explain how DNA is replicated. The DNA is made up of two strands, and first you have to break the two DNA strands. And this is done by separating or breaking the hydrogen bonds by an enzyme called DNA helicase. And DNA helicase unwinds and unzips the two DNA strands when it breaks those hydrogen bonds. Once you've got those two DNA strands separated, you have to join 3 nucleotides to the DNA strands, and that is done through complementary base pairing. The hydrogen bonds between the 3 nucleotides and the nucleotides of the DNA strand. And this is done by an enzyme called DNA polymerase. Now those 3 nucleotides are separate to each other, so they also need to be joined to each other. So to join sections of adjacent DNA nucleotides together to form the phosphodiester bonds between the phosphate of one nucleotide and the deoxyribose sugar of an adjacent nucleotide, that occurs using an enzyme called DNA ligase, which you would have heard about in genetic engineering. Now this was a famous experiment done by Messerschmitt and Stahl, where they looked at how DNA replication occurs. And if you start off with 2 strands in DNA, the question is, when making a new DNA molecule, does the 2 old parent strand stay with each other, or do they separate and you end up with two DNA molecules that contains one old parent strand and one new daughter strand? And in the experiment they started off with bacteria containing DNA that they use nitrogen-15. So all the DNA contained nitrogen-15. And then they put the bacteria in a medium containing nitrogen-14 to see what sort of nitrogen the DNA would have. And one idea is that if the old parental strands stayed with each other, that means half of the DNA will contain nitrogen-14 and half of the DNA will contain nitrogen-15. And this is known as conservative replication. The alternative idea is semi-conservative replication, and this is where you start off with nitrogen-15 DNA, and if both parent strands then separate and you add DNA nucleotides containing nitrogen-14, you'll have 100% of DNA molecules containing one old parent strand and one new daughter strand containing nitrogen-14. So in other words, every single DNA molecule will contain half nitrogen-14 and half nitrogen-15. So after one replication, the double-stranded DNA molecule containing only nitrogen-15 would therefore disappear. There would be no more DNA molecules containing nitrogen-15 in the semi-conservative replication, which is the second example. Whereas in the first example, if it was a conservative replication, that means you'd still have DNA molecules that would have nitrogen-15 100% in the whole DNA molecule. And what they found through these experiments is after one replication, all the DNA had a density halfway between nitrogen-14 and nitrogen-15. Therefore, DNA must replicate through semi-conservative replication, which is the second example. So this is proof that when DNA replicates, the two parental strands separate and then DNA nucleotides are free, joined to each one to form newly synthesized daughter strands. So every DNA molecule will contain one old parent strand and one newly synthesized daughter strand. Here we've got an example of DNA on the left side, but we've also got examples of RNA, in this case both mRNA and tRNA. So what is the difference between DNA and mRNA/tRNA? The DNA has got deoxyribose pentose sugar, where the hydroxyl on carbon 2 is replaced by a hydrogen atom, so the oxygen atom is missing. Whereas in mRNA and tRNA, there is a ribose pentose sugar instead. DNA is double-stranded, whereas RNA is single-stranded, and the DNA contains thymine nitrogen spaces, whereas in RNA, the thymine nitrogen spaces are replaced by uracil. In terms of similarities, both DNA and RNA contain phosphodiester bonds between the phosphate of one nucleotide and the pentose sugar of the adjacent nucleotide. Both contain guanine, cytosine, and adenine nitrogen spaces. So as we mentioned earlier, the difference is between thymine and uracil. And then in DNA and tRNA only, but not in mRNA, there are hydrogen bonds between complementary base pairs. So those hydrogen bonds form between the nitrogen spaces in DNA to link the two strands together, whereas in tRNA you've only got one strand, and in that one strand they can form hydrogen bonds with complementary bases with each other, and you can see it's got a strange sort of clover shape. But this doesn't occur in mRNA. So mRNA does not have hydrogen bonds, but it is a similarity between DNA and tRNA. Describe the nature of genetic code found in a gene. So if we look at a gene, what is the genetic code? And the genetic code is the order of bases in a gene. So if we look at this example here, we're going to try to read the order of these bases, and we can see that is T for thymine, then A, C, G, A, and A. So the order of bases provides the genetic code. It provides the information in a gene. And that information or instruction could tell the cell how to make a protein. So it's a bit like a recipe. The order of the bases, nitrogenous bases, could tell you how to then make the protein. It's a bit like when you put ingredients in order to make food. The order of nitrogenous bases in the gene can then tell you how to make the protein. In other words, which amino acid you put first and then which amino acid you put next until you put them into a sequence to make the protein. As I saw earlier, there are 20 different types of amino acids. All of these amino acids are similar to each other because they have got an amino group made of NH2, a carboxyl group, COOH. They've also got a hydrogen, but they're different to each other due to the R group, which can be polar, non-polar, or ionic. And proteins are different to each other. If you look at the example here, you could see that the amino acids are in different order between protein 1 and 2. So they can be made of the same amino acids, but the order of amino acids that you put into a sequence means that they are different proteins. So in the example of protein 1, you've got amino acid 1, 2, 3, 4, and then it's 2. And that would be the genetic code. So each of those amino acids represent an example you showed in the previous slide. Whereas in protein 2, you've got amino acid 3 first, then 1 again, then 2 and 4. So you could use the same amino acid, it's just the sequence of how you put these amino acids in order is different. And that how you determine that is instructed through the genetic code. In other words, the information in the DNA will tell you how to put which amino acid first and which to put second, and so on. The different sequence of amino acids make different proteins. Now, when you're making proteins or synthesizing proteins, which is protein synthesis, there are two stages. There's transcription, then translation. In transcription, you start off with your genetic code, which is the sequence of bases, the order of bases in the gene, and then you make a copy of that, and that copy is called mRNA. And you can see in the mRNA, you've still got the genetic code, you've got the sequence of bases. So the whole point of transcription is to make a copy of the genetic code, but this copy is called mRNA. Then that copy, which has the genetic code, which is the instructions, can be used to put amino acids in order to make your polypeptide, which is going to form the protein. And that happens in translation. So therefore it tells you or gives you the instruction on how to sequence the amino acids in the correct order to make the protein that you want. And then you join those amino acids to each other, as we said earlier, using peptide bonds in a condensation reaction between the carboxylic group of one amino acid and the amino group of another to form the polypeptide. So here's an example of how genes can be different to each other. If we look at the slide here, imagine that there are two different genes here, a protease gene and a lipase gene. Both of these genes are different to each other because their genetic code is different. In other words, the order of bases in the gene is different to each other. You can see that the genetic code in protease, for example, is ACCAAA. In real life, that would be longer, but we're just looking at a short example. Whereas in lipase, how is the lipase gene different to protease? You can see that its order of bases, CCGAGT, those sequence of bases are different. So it has a different genetic code. And then after you make a copy of it, which is called mRNA, we then use that instruction, that genetic code, to put amino acids in different order. So you can see that the amino acid that we're putting in order is different in protease in comparison to lipase. And so by having amino acid sequence in different order, you make different proteins. So different sequences of amino acids therefore make different polypeptides, because we know that those amino acids will have different R groups. So that means they'll have different properties. They can form different bonds, different secondary structures, different tertiary structures, and possibly different quaternary structures. So therefore they're going to fold differently and make different proteins. And because of the way they fold, they could then form proteins that have got different functions, as we saw in the example of collagen and hemoglobin. So the sequence of amino acids in a polypeptide chain determines the specific shape, and clearly that shape will determine the protein, whether it's a fibrous protein or a globular protein, and then what sort of function that protein will therefore have. So examples of that is enzymes with different shape. Active sites will have different functions, or you could have proteins on the cell surface which have different antigen shape, or even lymphocytes producing different antibodies which have got different shapes as well, therefore different functions to attack different pathogens or attach to different antigens and pathogens. As we mentioned earlier, the amino acids you put in order is really important, because the way they then fold into a secondary, tertiary, quaternary structure would therefore have an impact on its function. So what were the stages of protein synthesis? We said that there was transcription and translation. So if we look at this gene here, a gene is realistically just a sequence of bases on a DNA molecule. That sequence of bases is the DNA genetic code, because it gives you the instruction to put amino acids in order. In transcription, you're actually making a copy of the DNA genetic code called mRNA, and then that mRNA codes for, you can see here, if we start off with the first three bases, AUG, it codes for a MET amino acid. The next one is ser or serine, leucine, and so on. The UGA codes for stop. That's when you know when not to add any more amino acids. So a gene is a sequence of bases on a DNA molecule that codes for a sequence of amino acids in a polypeptide chain. And that is the whole point of protein synthesis, to use the instruction of a gene, the genetic code, using that instruction to put amino acids in order to make the polypeptide. So explain the nature of genetic code. The genetic code is made up of base triplets, which is known as codon. So when you read the mRNA, it's actually read in every three bases together. So the first three bases or base triplets or codon that I read was AUG, and each of these codon codes for a specific amino acid. So AUG codes for the amino acid MET. Now if you look at how you read these codons on the genetic code, the first six bases is AUG UCA, and you'd read them AUG as the first codon and UCA as the next codon. You wouldn't be AUG, then you skip the first nitrogenous base, and which means the next one be UGU and then the next one be GUC. Okay, that's wrong. It's first AUG, not AUG, UGU, GUC. That's wrong. It should be just AUG, then UCA. So in other words, the genetic code is non-overlapping, and that's how the ribosomes read them. They read them codon at a time, where it's three bases or base triplets first, and then it's the next base triplet is one codon. They are non-overlapping. Now when you look at the example here, you've got the leucine amino acid can be coded by 4 different codons. So this means that amino acids can have more than one codon. We call that degenerate code. So the genetic code is degenerate because more than one codon can code for the same amino acid. And you can see the difference between those codons for leucine is the third base in the base triplet. It could either be U, C, A, or G in this case, and different amino acids will have different number of codons that can code for it. Now, when the ribosome attached to the mRNA only reads from the start codon to the stop codon. So the start codon is always AUG, and the stop codon, there are 3 different types that can be used, and that's when you stop reading them or translating to form the polypeptide chain. And not all of the genome or DNA codes for protein, so some parts of it is non-coding. So if the ribosome reads from the start codon to stop codon, the DNA before the start codon or RNA before the start codon, and the same with the stop codon afterwards, all that basis there is non-coding. It does not code for an amino acid. Explain the process of transcription in nucleus. So here we've got our DNA molecule, and first you have to unzip the DNA by breaking the hydrogen bonds, and that's done by an enzyme called helicase. This to produce mRNA, which is a copy of the DNA genetic code. It's a copy of the sequence of bases or the bases order. We want to know what the role of the sense and antisense DNA and mRNA is. So the coding strand, the strand that contains the genetic code, the gene, is the sense strand. The opposite strand, which is known as the template strand, is the antisense strand. And what you do is to make the mRNA, you get 3 RNA nucleotides. They are joined to the template strand using complementary base pairing, and that obviously requires the use of hydrogen bonds. And this is done by an enzyme called RNA polymerase. So RNA polymerase gets 3 RNA nucleotides to hydrogen bond with complementary base pairing to the template strand, which is the antisense strand, that is the opposite of the coding strand, the sense strand of the gene. And then you join the adjacent 3 RNA nucleotides to each other via phosphodiester bonds. Again, this is done by RNA polymerase. Once that mRNA is made, that mRNA, as we said, has genetic code, which has got the same instruction as the DNA, with the difference between the DNA and the mRNA being what we mentioned earlier in terms of the difference between nucleic acids. And once that mRNA is made, it leaves the nucleus to go to the cytoplasm, where it attaches to a ribosome or a ribosome in the rough endoplasmic reticulum. So here's another example. You've got your DNA strand. It contains the gene, which is the coding or sense strand. The opposite strand is the template or the antisense strand, which is non-coding. The first thing you do is you break the hydrogen bonds between the 2 strands, and then you add 3 RNA nucleotides using complementary base pairing. So we can remember this with GCAT, but because it's RNA, there's no thymine, so the next base would be G, C, but instead of T, it'd be U, because uracil replaces thymine in mRNA, and so on to form the mRNA. And you can see this mRNA is complementary to the template strand, and you form phosphodiester bonds between the RNA nucleotides, which is done by RNA polymerase. And then when the mRNA is formed, you could see that that mRNA has the genetic code as the gene, the coding sense strand. It's got exactly the same instructions that we could then use as an instruction to make or synthesize the protein. That mRNA can then leave the nucleus to make or synthesize protein. Okay, describe the structure of mRNA. So what is the structure of this mRNA, including its nucleotide? We can see that it's obviously got a sugar phosphate backbone, and then between the phosphate of one RNA nucleotide and the ribose sugar of an adjacent RNA nucleotide, there is phosphodiester bonds. So as we mentioned the structure of DNA earlier, the structure of mRNA is the same except for differences such as the fact that it is a single strand. It still has phosphodiester bonds. There are ribose sugar instead of deoxyribose, and in terms of bases they are all the same, but thymine nitrogenous base is replaced by uracil in mRNA. How about the structure of tRNA and what is its role? If you look at tRNA here, as we mentioned earlier in terms of its structure when comparing it to DNA and mRNA, the tRNA has bonds between its RNA bases which are complementary, and that gives the tRNA a clover shape due to the hydrogen bonds that form between the complementary RNA bases. Explain process of translation at ribosome. So first the ribosomes attach to mRNA, and they only read from the start codon, which is AUG, to the stop codon of the mRNA. Now each tRNA transfers an attached specific amino acid to the mRNA. And there are lots of different types of tRNA. So how do we know which tRNA should transfer which specific amino acid? How do we know if it will transfer the correct amino acid in the correct specific order? This is done because the tRNA anticodon is complementary base pairs with the mRNA codon using hydrogen bonds. So each tRNA contains not only a specific amino acid, they also have 3 very distinctive anticodons which has to complementary base pair with the mRNA codon for it to be the correct tRNA. And then that correct tRNA will transfer the correct specific amino acid. So in the example here, we can see that AUG, which is the codon on the mRNA, codes for the amino acid MET. So we need a tRNA that will transfer the MET amino acid. How do we know that the correct tRNA will transfer the MET amino acid? That's because it contains the complementary tRNA anticodon UAC. So only a tRNA with UAC anticodon will transfer or come along and transfer the specific amino acid MET. And then in the next example, you could see that ARG amino acid is transferred. And by continuously transferring the correct specific amino acid in the correct order, you can make a polypeptide chain by joining these amino acids through a peptide bond. And this makes the polypeptide chain, which can then form into a secondary, tertiary, and or quaternary structure that will determine its function. So here you can see the example. We've got our mRNA containing codons, and the ribosome first attaches to the mRNA, and it will attach and start on AUG, which is the start codon. When it attaches to the start codon AUG, a tRNA comes along, which has got the complementary tRNA anticodon to AUG, and that tRNA will bring the specific amino acid MET. And then you do the same for the next mRNA codon and so on, and then these amino acids are then joined to each other through peptide bonds to make the polypeptide chain until you reach the stop codon of the mRNA. Explain the term point or gene mutation. A point-stroke gene mutation is when there's a change in the base sequence. And if the base sequence changes, that means the order of bases has changed. So therefore, you've got a different genetic code. So that means the instruction to make a polypeptide or how to put the amino acid in the correct sequence is going to be different. And there are 3 different types of point mutations. So here we've got our first example, and if you compare the 2 genetic codes, you could see in the second one that there is a cytosine base missing. So this is an example of a base deletion, and when you delete a base, that shifts every other base, which therefore has an effect on every other amino acid that is coded for. Here's another example of a point mutation. So in this example, we've got a guanine base that's been inserted. So this is known as a base insertion. And then the last example, you can see that the cytosine is substituted for a thymine nitrogenous base. Every other base is still the same. So this is known as a base substitution. Discuss possible consequences for amino acid sequence due to point mutation. So with these 3 different types of point mutations, what is the consequences to an amino acid sequence? What effect will it have on the polypeptide? In the base deletion and addition, you could see that it shifts every other base, and this is known as a frame shift, which has a big effect on which amino acid is coded for. So you end up with different amino acids that are coded for after the frame shift due to a base addition or deletion. However, for base substitution, here an amino acid can be replaced by another amino acid, or sometimes there is no effect in the mutated codon because the substitution in the codon causes a codon that codes for the same amino acid. And this is due to degeneracy of the code, because more than one codon can code for the same amino acid. So sometimes there is a base substitution, but it still codes for the same amino acid. So there is no effect. And this happens on the 3rd nitrogenous base of a codon in certain amino acids that are coded for. Now, if you have a frame shift or the amino acid substitution codes for a stop codon, that means you end up with a shorter polypeptide chain. And this is because translation will stop sooner and no more amino acids will be added. So that is another effect on the amino acid sequence that could happen. Now, the overall effect of this is that different amino acid sequence, in other words, you have a different primary structure now, and because you end up with a different primary structure or amino acid sequence, that means that there are different amino acid sequence with different R groups, and this results in changes to the possible types of ionic, hydrogen, and disulfide bonds that could happen, and due to the different types of bonding, it alters the folding of the polypeptide chain. So you could end up with a different secondary, tertiary, and or quaternary structure of the protein. So you can see there are huge possible changes due to point or gene mutations. And this slide shows an example of those possible changes that could happen and how the amino acid sequence change could therefore form different bonds due to different R groups, and therefore they could form different shapes due to the way that they fold differently. Explain effect of point mutations on amino acid sequence as illustrated by sickle cell anemia. So this is a good example that you need to know for your exam. So here is the point mutation in sickle cell anemia, and if we spot it, we could see that a thymine nitrogenous base is being substituted into an adenine. So thymine in DNA point mutation is being replaced by adenine. This is a base substitution point mutation. So instead of GAG in the mRNA, we've got a GUG mRNA codon, and that GUG mRNA codon complements a tRNA anticodon that carries a valine amino acid instead of glutamic acid. So we end up with valine instead of glutamic acid in the polypeptide chain on the 6th amino acid. And this single amino acid chain, having valine instead of glutamic acid, results in a sticky, rigid beta-globin polypeptide chain. Therefore, hemoglobin is less efficient in carrying oxygen to tissues, and because it's less efficient at carrying oxygen, that means that this leads to somatic cells, body cells receiving less oxygen, so therefore less energy can be released in respiration, so there's going to be more tiredness, which is known as anemia, and that's why it's called sickle cell anemia. And it also causes the red blood cells to become sickle-shaped. And because they're sickle-shaped, they increase the chance of blocking small blood vessels due to their shape. Again, that's red blood cells less likely to transfer oxygen to the somatic cells, and there's an increased chance of blocking these small blood vessels due to the sickle shape of the red blood cells. Explain how point mutations can bring about change in structure and activity of enzyme. So in this example here, we're looking at point mutations that now affect enzyme. And so if you have a change in the sequence of amino acids, as we said earlier, that affects the primary structure, you end up with a different R group. And that different R group means that you can now form different ionic hydrogen and disulfide bonding. Those bonds are important in the formation of tertiary structure. So therefore, a mutation, a point mutation, could result in a change in the enzyme's active site shape. In other words, how specific that enzyme is doing in its function. And the substrate will no longer bind to the active site, and so therefore you can't have any more enzyme substrate complex being made. And that's something we're going to discuss on our next topic of enzymes. Explain how enzymes work. Enzymes are biological catalysts. They're catalysts, which means that they increase the rate of reactions, and they're biological catalysts because they're found in organisms. They can catalyze intercellular reactions or extracellular reactions. So intercellular reactions are those reactions that happen inside cells. An example of an enzyme that does this is DNA polymerase, which is involved in DNA replication. Extracellular reactions is when you secrete or work outside cells, such as enzymes involved in digestion. Lysozyme enzymes is an example as well. All enzymes are globular proteins because they have a specific shape due to their primary, secondary, tertiary, and often the quaternary protein structure. So how do enzymes increase the rate of reaction? It's because enzymes decrease the activation energy of metabolic reactions, thus decreasing the reaction time, and this increases the rate of reactions. Here's a diagram showing how the enzyme and substrates bond with each other. The important part of the enzyme is its active site, and this active site has a specific complementary shape to fit the substrate, and when it does that, that lowers the activation energy by forming an enzyme substrate complex. So when the substrate bonds to the enzyme's active site, it forms an enzyme substrate complex, which lowers the activation energy to increase the rate of reaction. In GCSE, you learned about the lock and key theory, but in A level, we learn about a slightly different idea called the induced fit theory or the induced fit model. In this case, there's an interaction between the enzyme active site's R group and the substrate, which conformationally changes the active site shape. And when it does that, this induced fits the substrate binding into the enzyme's active site to form the enzyme substrate complex. So in the diagram, you could see that the active site doesn't necessarily have exactly the same complementary shape as the substrate, but when they start bonding or bonds form between them, the substrate then causes a change in the enzyme's active site's shape to induce fit the substrate binding onto the enzyme's active site, and that forms the enzyme substrate complex. So we know that when the substrate fits into the enzyme active site, that forms the enzyme substrate complex. But what happens if the shape of the active site changes? And we've learned from GCSE this is called denaturing. Other ideas we learned from GCSE is when you increase the temperature of molecules, they have more kinetic energy, so they move faster. In denaturing, we also know that from what we've learned in A level, these enzymes have got a secondary, tertiary, and possibly a quaternary structure due to the bonding that could form. That could be obviously ionic bonds or hydrogen bonds and or disulfide bonds. And these bonds can be broken and new bonds being made due to a change in temperature. And that's what happens when an enzyme's active site denatures. Explain how initial rate of enzyme activity can be measured and why is it important to measure the initial rate of an enzyme's activity. So here's a graph showing how enzyme activity changes over time and why do we measure the initial rate. And the rate of reaction can be measured by calculating the gradient, which is just the change in y divided by the change in x. So enzyme activity divided by time. And why measure the initial rate of enzyme? This is done when the substrate is in excess, so not limiting. If you were to measure the rate of enzyme activity at other times, not the initial time, the problem would be that the substrate might start to run out. It would not be in excess, so it would not be a fair comparison between different enzymes. So you want to make sure that there's enough substrate so you can make a comparison on the enzyme activity rate. And this is done at the initial rate. Explain how different factors affect rate of enzyme activity. So we should know 3 different factors. There's temperature, pH, and substrate concentration. Obviously, a 4th example would be enzyme concentration as well, but these are the 3 that you specifically need to know for A level. And for temperature, if we look at A in the graph, you can see that when you increase the temperature up to the optimum temperature, this increases the rate of enzyme activity or the rate of the reaction. We know this because the enzyme and substrates move faster because they have got more kinetic energy when you increase the temperature. Therefore, there'll be more successful collisions between the enzyme's active site and the substrate. And when there's more successful collisions, therefore more enzyme substrate complex will form. And that's where you get an additional mark in the A-level exam when you mention that. At B, you can see that the range of enzyme activity is at its highest. And this occurs at the optimum temperature. Whereas at C, the increase in temperature above the optimum means that there's a decrease in the rate of enzyme activity or the rate of reaction. And we know this because the enzyme active site denatures. There's a change in the tertiary structure due to the temperature being too high. And this causes a disruption in the ionic or hydrogen bonds of the enzyme active site. And when that happens, the active site changes shape, so no longer fits or binds to the substrate, and therefore you have no enzyme substrate complex forming, thus decreasing the rate of enzyme activity when you increase the temperature above the optimum. pH is slightly different because first, if you look at E, you could see that the rate of enzyme activity is at the highest at the optimum pH. However, if you were to decrease or increase the pH from the optimum, so it doesn't matter which direction it is, whether you decrease the pH, which makes it more acidic, or you increase the pH, making it more alkaline, or less acidic, you then decrease the rate of reaction, the rate of enzyme activity. And this decreases the rate of enzyme activity as mentioned, and we know this because this causes the enzyme to denature. There's a change in the tertiary structure. In this case, it's not due to too high a temperature. It's because when you change the pH, you're changing the hydrogen ions that are involved. You're changing how acidic it is. And that acidity or hydrogen ions could disrupt the ionic or hydrogen bonds of the enzyme active site, and therefore the active site changes shape, so no longer fits or binds to the substrate, and no enzyme substrate complex forms, and this decreases the rate of enzyme activity. Substrate concentration is slightly different. So you can see at G, when you're increasing the substrate concentration, that increases the rate of enzyme activity because there'll be more successful collisions and therefore obviously more enzyme substrate complex forming. However, at a certain point, such as H, the maximum rate of reaction stays constant, and that's because all the active sites are full. So there cannot be any more enzyme substrate complex forming because the substrates are already binded to the enzyme active site. You can't bind any more substrates. It's at its maximum rate, so the maximum rate of reaction stays constant at very, very high substrate concentrations. Describe how enzyme can be affected by inhibition. There are two types of inhibitors you've got to know: competitive inhibition and non-competitive inhibition. And the difference between them is that in competitive inhibition, the molecule has got a similar shape to the substrate, and therefore it could also bind to the enzyme's active site. And due to this, that means it could prevent the substrate from binding, which means that there's going to be less enzyme substrate complex forming, thus decreasing the enzyme activity and the rate of reaction. However, in non-competitive inhibition, the molecule binds to a different location of the enzyme. So we call this location the allosteric site. And because it binds to the allosteric site of the enzyme, it could disrupt the bonds that make up the globular structure of the enzyme, such as disrupting the ionic bonds or the hydrogen bonds or the disulfide bonds of that globular structure. And that means that when you change the enzyme's bonding, you also change its active site shape. So substrates can no longer bind to it, and this means that you can't form enzyme substrate complexes. And this still occurs when the substrate concentration is very high, because even if the substrate concentration is very high, by having non-competitive inhibitors, they change the active site of the enzyme. So it doesn't matter if you have more substrate, they can't bond with the enzyme's active site, and therefore the enzyme activity decreases. However, in competitive inhibition, if you increase the substrate concentration, that means that they're more likely to outcompete the competitive inhibitor, so therefore you can increase the rate of reaction even if there is competitive inhibition, because there's going to be more substrates. So that is one difference, or how you could tell the difference between a competitive inhibitor and a non-competitive inhibitor. A competitive inhibitor can be overcome by increasing the substrate concentration to a very high concentration, whereas a non-competitive inhibitor, the substrate concentration will not have any effect because you'll change the non-competitive inhibitor, when it binds to the allosteric site, changes the enzyme's active site shape, so it doesn't matter if you have more substrate concentration. One other thing to understand is that in a non-competitive inhibitor, the reaction doesn't reach the maximum rate because the amount of enzyme activity you could have decreases due to the less chance of enzyme substrate complexes forming, as more of the enzyme active site will have changed in terms of its shape. However, in a competitive inhibitor, it can still reach the maximum rate when you're increasing the substrate concentration, as long as it can overcome the amount of competitive inhibitors molecules. Now, this is important in reactions because a lot of reactions that happen in our body are not necessarily one reaction, they're actually a series of reactions. So an example here, you can see you've got a substrate to start off with A, you've got an enzyme that binds onto that substrate, that forms the product B, and then you've got a different enzyme that is involved in that reaction to convert it to the product C, and then a different enzyme converts it to a product D. So you've got a series of reactions here with 3 different enzymes. And in terms of end product inhibition, the end product inhibits one of the original enzyme that catalyzes an earlier reaction. So for instance, an example would be here, an end product inhibits enzymes involved in earlier reactions of a series of reactions. And this helps regulate end product quantity via something called negative feedback. So to help you understand this, if you had too high product D, so you produce too much product D, that product D could inhibit the enzyme activity of enzyme 1. So in other words, it will inhibit enzyme 1 activity, therefore less product B would be produced. If you produce less product B, that means less product C would be produced, and that means less product D would produce, therefore decreasing the production of D. If the D molecule concentration decreases a lot, that means there'll be less inhibition of enzyme 1, and that means you can make more product B, and if you got more product B, you can make more product C, and therefore you can make more product D. So that's an example of negative feedback, where if you have too much of molecule D, it inhibits enzyme 1, which means that you end up making less product D. But if you have too low product D, it inhibits enzyme 1 less, so you end up with more product D, negative feedback, thus decreasing the production of D. So core practical 1 is where you investigate a factor affecting initial rate of enzyme controlled reactions. So we should know different factors that could affect enzyme activity. So here you've got to know about the different types of variables. So factors that could affect it would be temperature, pH, substrate concentration, and also obviously enzyme concentration as well. So the dependent variable is the thing that you're measuring, the independent variable is the one that you are changing, and then control variables are things that you keep the same. So it's really important, whichever factor you're doing, the one that you change, you then have to find a way of measuring it, dependent variable, and the other variables have to be kept the same, they'd be control variables. If you are looking at how pH affects the initial rate of enzyme reaction, remember it's got to be initial rate because the substrate has to be in excess to make a fair comparison. You could use a pH buffer solution, so you keep the pH the same to test out on that pH, and then you'd use a different pH for subsequent reactions to compare how pH affects the initial rate of enzyme reaction. All the other factors would therefore be control variables, so you'd have to keep the temperature the same. This could be done using a water bath. The enzyme and substrate concentration will also have to be kept the same, but you'd put them in excess so they're not a limiting factor. If you changed it and the independent variable was temperature, again you could have temperature in a controlled using a water bath, so you'd use different water baths to accurately measure the temperature, and this time around you'd keep the pH the same as the control variable and the enzyme and substrate concentration. Whatever you change in terms of the independent variable, you want to have at least 5 different types of independent variable. So if you did temperature, you'd want 5 different temperatures at a range. So for example, it could be something like 30 degrees Celsius, then 35, 40, 45, 50. So you'd have 5 different examples of your independent variable, which you should note down in an exam answer. This could be the same for pH or enzyme concentration or substrate. Always mention what the control variables are and how you'd control them, and then don't forget to mention how you'd measure the rate of reaction as well, the initial rate of reaction. Describe why investigations are repeated. When you repeat an investigation, it gives you the opportunity to calculate a mean, which is an average. You could also calculate a standard deviation or standard error. It also helps you to recognize anomalies. So you can spot anomalies when you do repeats. You should do at least 3 repeats. That way you can see which one's an anomaly in comparison to the other 2 repeats. Why is it important to look at standard deviation or standard deviation bars when you plot them on a graph? Why is this significant? The standard deviation can show variation, or they can show you what the range of results are or how the data is spread in comparison to the mean or the average. They give you an indication of how reproducible or repeatable the investigation or the practical is. And then if there's any overlap, it gives you an indication of how significant these differences are.